Okay, so welcome to this next video in which we are discussing defects in excitation contraction coupling in hypertrophied cardiomyocytes. So we've discussed now the pathway that leads to pathological remodeling of cardiomyocytes. Okay, so originally something is stopping the heart from being able to perform its function. Okay, it leads to the pathological hypertrophy of the cardiomyocytes in the left ventricle. And I want to stress as well now that it's not a case that all of the cardiomyocytes are suddenly going to undertake pathological remodeling. It will be a process that occurs over a long period of time, and gradually more and more of them are going to be converted from the normal form to this hypertrophied form. And then these hypertrophy forms, temporarily they're capable of generating a larger amount of force. But then, gradually, over a period of time, their ability to generate force goes down, and it goes down below what they were capable of generating prior to the pathological remodeling. And as more and more of the cardiomyocytes in the heart go to this pathologically hypertrophied form, then the force that the left ventricle is capable of producing goes down and down and down. That means that the amount of blood the left ventricle is going to be ejecting into the aorta goes down and down. So the cardiac output, the amount of blood it ejects in a minute, goes down. And once that crosses the threshold where it's no longer capable of pumping enough blood from the venous to the arterial side to meet the needs of the body, then you've got heart failure or cardiac failure. Okay, so cardiac failure is when the heart is too weak to pump enough blood around the body in a minute to meet the needs of the body. So the heart is failing the rest of the body. Right, okay, so let's now discuss why in pathological cardiac hypertrophy these cells lose the ability to contract. They lose the force that they generate when an action potential stimulates them to contract. Okay, so we've discussed in previous videos how there are defects in the circuit 2 pump in these hypertrophied cardiomyocytes, and that leads to too little calcium in the SR, so that when you come to release the calcium during contraction, then you get a smaller release of calcium, and therefore you recruit fewer sarcomeres to actually contract. So the number of sarcomeres contracting in the cardiomyocyte goes down, and that means that the force with which the cardiomyocyte contracts also goes down. Now what we're going to look at is defects in excitation contraction coupling. So let me just remind you of how excitation contraction coupling works within a cardiomyocyte. Okay, so let's say we're working with a ventricular cardiomyocyte, which means that they have these T-tubules. And it's worth noting that in atrial cardiomyocytes, the T-tubules are not present. Okay, so here is our ventricular cardiomyocyte. Right, so when this cardiomyocyte gets stimulated by an action potential, what will happen is the action potential will spread along the membrane of the cardiomyocyte, and it will activate channels in the membrane of the cardiomyocyte. Okay, and these are voltage-gated calcium channels of the L type. So this here, this is an L-type voltage-gated calcium channel. Okay, and these channels are also often referred to as dihydropyridine receptors because of their sensitivity to the dihydropyridine drugs, so an example of which is nifedipine. So, a few abbreviations here. VGCC is short for voltage-gated calcium channel. DHPR is short for dihydropyridine receptor. I suppose I should write out the name dihydropyridine receptor in case you haven't seen it before. Dihydropyridine receptor. Okay, right. So, this is a voltage-gated calcium channel. So, when the action potential arrives at this portion of the membrane, it will result in depolarization of the electrical potential difference across that membrane, which is going to lead to the activation of this L-type voltage-gated calcium channel in pink here. Okay, so the L-type voltage-gated calcium channel is going to open, and it will allow calcium to enter from the outside of the cell into the cytoplasm of the cell. Okay, right, so what does this calcium do next? Well, it acts on receptors that are in the membrane of the sarcoplasmic reticulum. Okay, so you will have a process coming off from the sarcoplasmic reticulum, which is an intracellular membrane-bound organelle, and this will have a membrane that is interfaced with the membrane of the cell, like so. So this is a picture of the sarcoplasmic reticulum here, with these many processes which form these synapses, as they're called. So this sort of whole structure here, this is called a calcium synapse, for good reason. I'll explain this in a moment. Okay, so if you think about synapses between two neurons, what you have is a presynaptic membrane which releases neurotransmitter. This plasma membrane is the equivalent of the presynaptic membrane, and the calcium is the equivalent of the neurotransmitter. In the case of the neurons, the neurotransmitter diffuses across the synaptic cleft. In the case of the calcium synapse, the calcium will diffuse across this dyadic cleft. So the gap between the plasma membrane and the sarcoplasmic reticulum membrane is known as the dyadic cleft. Okay, so calcium will diffuse across the dyadic cleft, and it will bind to receptors in the sarcoplasmic reticulum membrane. Okay, so the sarcoplasmic reticulum membrane is then the equivalent of the postsynaptic membrane. So here is a receptor in the postsynaptic membrane, and the type of receptor that is in the sarcoplasmic reticulum membrane is a type 2 ryanodine receptor. Okay, so these type 2 ryanodine receptors are going to act upon stimulation by the calcium that is diffused across the dyadic cleft. Okay, so let me colour in the type 2 ryanodine receptor in turquoise. Right, so basically the sarcoplasmic reticulum is filled with calcium. Okay, it's got a very high calcium level compared to the cytoplasm. So when you open the type 2 ryanodine receptors upon stimulation by the calcium, calcium will leave from the lumen of the SR and go out into the cytoplasm of the cell. Now, basically, you might ask, surely the calcium, by the way, is the thing that causes the contraction of the cardiomyocyte. But you might ask, well, why do we need this? Surely we can just let this calcium come in from through the L-type voltage-gated calcium channels, and that will be enough. But the reality is hardly any calcium comes in through these L-type voltage-gated calcium channels. Okay, so the amount of calcium coming in this way is tiny, and that little calcium signal that comes in through the L-type voltage-gated calcium channels, it has a name. It's called a calcium sparklet. So the little bit rise in calcium that you get around an L-type voltage-gated calcium channel when it opens is called a calcium sparklet. The calcium sparklet is going to diffuse across the dyadic cleft and open the type 2 ryanodine receptors, which will release a huge amount of calcium from the SR. And this much bigger rise in calcium that you get because the type 2 ryanodine receptor is opened, that's called a calcium spark. Okay? So at each of these calcium synapses, what you'll have is calcium sparks being produced. The calcium spark that's tiny, but the calcium spark is bigger. Okay, so it's the calcium sparks that are going to actually cause contraction. These are too small. They'll contribute, but hardly anything, basically, trivially. So this is the major cause of contraction. So you'll have multiple of these calcium synapses all over the place, and all of them will be producing calcium sparks. So you'll get calcium going up at all of the calcium synapses, and the summation of all of the calcium sparks together is what's known as a calcium tide. So the global rise in calcium that you get within the cytoplasm of the cell, which is all of the calcium sparks summated together, is called the calcium tide. Okay, and this calcium tide will then cause the sarcomeres to become activated and they'll start contracting. Okay, so what do we find goes wrong with this excitation contraction coupling process in hypertrophied cardiomyocytes? Okay, well basically what we find is that the calcium tides that you get are smaller. Okay, so when an action potential arrives on a cardiomyocyte, it will produce a certain amount of calcium release in the cytoplasm. I.e., you'll get a certain level of calcium tide. So if you measure concentration of calcium at any point of your choice, it will look something like this. So if I draw a graph, if we have calcium concentration versus time, okay, then when an action potential occurs, what you'll get is something that looks like this. Calcium will go up and up and up and up, okay? And it will reach a certain maximum, basically, and then it will come down again, so you'll get a spike like that. Now, in cells which have undergone pathological cardiac hypertrophy, this spike does not get up nearly as high. So the calcium tides are smaller. What you also see is that you're getting calcium sparks occurring even before you've actually stimulated with an action potential. So not only do you see that the actual calcium tide that you get when you do stimulate with an action potential is smaller, but you're seeing release of calcium at these calcium synapses before you've stimulated with an action potential. That shouldn't be happening, okay? So you see the calcium tide is smaller, and you see that you have calcium sparks which should not be there. Calcium sparks which are occurring even before an action potential has arrived. And if the action potential hasn't arrived, then you haven't stimulated the L-type voltage-gated calcium channels. So you haven't had the calcium spark to stimulate the L-type voltage-gated to stimulate the type 2 ranitidine receptors. So they should not be opening, but they open when they shouldn't, basically. So you find, if you just look at a cardiomyocyte that's at rest, that's undergone pathological cardiac hypertrophy, loads of calcium sparks are happening all the time that shouldn't be happening. You should only get calcium sparks happening when the action potential arrives, when it activates the L-type voltage-gated calcium channels, and the calcium spark that then activates the type 2 ranitidine receptor to release a calcium spark. They should not be happening just when the cardiomyocyte is at rest. But in hypertrophied cardiomyocytes, that seems to be happening. So, basically the response to an action potential is reduced, and the response to no action potential is increased, basically. So you're wasting effort releasing calcium sparks when there's no action potential, and when there is an action potential, the actual calcium tide that you get is smaller. And we'll discuss why this is in the next video.